My name is Sabah Anabulan. I'm from 8220 a. So today I'm going to be presenting about my slide presentation on propagation of oil palm, and this is slide presentation prepared for Dr. Amira Abdul Reza. Before proceed to my main content of this slide discussion, let's get to know the oil palm first. The oil palm is belong to the family of Arecaceae, called by scientific name of Elais guineensis, which originated from tropical rainforest region of West Africa. Why should we bother to know about this? It is because the oil palm has high oil content of the mesocarp and kernel of the fruit that are produced in a heavy bunch. As you can see on the diagram here, this is the Dura variety, Tinera variety, and Piscifera variety. In my discussion slide, I will emphasize on the Dura variety crossing over with Piscifera variety will produce the Tinera variety of oil palm. Let's watching the video on the simple step in oil palm seed production. So here is my main content for this slide discussion, is where I'm going to discuss on the method of propagating the oil palm, the pro and cons for each method, their importance, the economic impact, from using the each of the different method, and also the future development of oil palm. The oil palm propagation technique is by using the tissue culture and seed germination. For the tissue culture technique, which is also known as micropropagation or cloning, it is a method of producing identical or true-to-type photocopy of a selected or test by growing plants from the leaf tissue, which is the immature leaf, or efflorescence, and also by using the tip of tertiary roots of Tinera oil palm with desirable traits. The technique is started in 1960s, and in the middle of 1970 it is proven as a success technique for propagating the oil palm. Meanwhile, the next technique is the seed germination, is where the oil palm seed is dormant when harvested and germinates intermittently over several years in natural condition. The method is for germinating seed in the early year of plantation were often erratic and germination failure were common. But in 1962, the critical requirement for breaking the dormancy was maintained at a temperature of 30 to 40 degrees Celsius up to 80 days. The tissue culture process is started by selecting the palm orchids with desirable characteristic. Well, in this case, we are using the Tinera nuts by removal of explant from close to the growing point, approximately around 25 to 30 centimeter. Only then we are start doing our excision and inoculation of the leaf explant. After 6 months, the colors induction and differentiation stage is occur where the development of colors on explant rests on the nutrient medium in culture tube. After 9 months later, the embryonic formation is where this stage is what we call the embryogenesis. Right after 24 months after, the polyembryoid multiplication stage occur, and 2 months later, there is a stage where we call the shoot development stage, is where we need to removal of the embryos from the colors and the development of the plantlet with functioning root and shoot. 4 months later, later is where the root induction stage, and within 3 to 4 months later, the plantlet is transplanted and we need to perform the preliminary hardening off right before we are transferring the plantlet to the field nursery for adaptation to ambient climatic condition. As for the method for seed germination is divided into two methods, dry heat method and also the wet heat method. As for dry heat method, the heating of the seed to the moisture level that is not too low for germination, but the heat reaction is compromised. The Dura nuts are preferable compared to Tinera nuts due to the Tinera nut possess poor moisture content below 20% or above 26%. The moisture level during this method is not critical as all the germination occurs within a few weeks under the mean of soaked and reheated condition. But this condition is only applied to the ungerminated seed during the germination period and does not cause brown germ disease as it happened in the tissue culture method. In contrast, the wet heat method, the seed must be at the right moisture content. As for Dura, we need is for Dura 21 to 22%. As for the Tenera is 26 to 28% of moisture content. The, so the wet heat method is by soaking the seed in the water for 5 days and then drying it in the shades for about 2 hours before packing it in the polyethylene bags and placing it in 39 degrees Celsius inside a heating chamber. The seeds were then placed in a cold spot after 80 days and the moisture content maintained as before. The germination then flush will begin after chilling and should take approximately 15 to 5 days to get complete. The process of seed germination propagation of dry heat method and wet heat method is slightly the same, but the difference is where the dry heat method on the last stage is that it keeps the seed tightly in the polythene bag at room temperature but remove every 5 days interval of germinating seed. Where at wet heat method is where the initial stage is where the seed must be at its optimal moisture content for germination. 21 to 22% is for Dura and 26 to 28% moisture is for Tinera. According to the Oil Palm 5th edition written by Corley and Tinker 2019, Tinera seed can be germinated by dry heat method, but the germination rate is poor, but successfully germinated using the wet heat method. Besides, they also recommended of moisture of 20% for heat treatment and 26% for germination. As for the advantage and disadvantage of the tissue culture propagation is that the new plantlet can be cultivated in a short period. The process is not seasonal and can be carried out at any time of year. On a large scale, the tissue culture technique aids in the delivery of novel subspecies and variety to the consumer market. In contrast, it required more workforce and high costing to conduct the techniques. High chances of the new plants being exposed to a disease and abnormality in flowering, bunch and truncated leaf syndrome (TLS). And last but not least is that while the success rate is high, when the proper protocol are followed, tissue culture success is not guaranteed. Meanwhile, for the seed germination propagation, the advantage is that it is one of the effective methods on propagation in any type of plant in the world. Genetic variation forming hybrids in vigor state increase with or then superior qualities. The plant seed can be stored for long period without losing its valuability. And last but not least is that one of the cheapest cost to conduct the technique does yield many trees. In contrast, the disadvantage is that oil palm takes the longer time to produce seed, which is the dormancy period, compared to the other cheapest technique in the world. Next is it is possible to produce a plant that does not identical to its parents. Makes sense. The last but not least is that the germination period is quite long just before it transferred to the pre-nursery and leads to long time to get matured. As for the importance of the tissue culture has a wide range of applications that can be used to reproduce the wide range of species. The plant genetic makeup can also be changed to make it immune to specific illnesses and viruses. Thus, the businesses and people will frequently propagate plants by using a tissue culture that contains specific features and more profitable to their business and appealing for personal usage. Meanwhile, the seed germination importance for some plants that can only be propagated by seed as if they are difficult to germinate, their continued survival is jeopardized. The seed germination determines the level of the plant output required for human existence in terms of medicinal plant, animal grassy feed, and also the food security. But bear in mind that the seed germination depends on factors such as a moisture percentage, temperature, light, and oxygen. The economic impact of propagating by using a tissue culture versus the seed germination is that the tissue culture costing in terms of production cost as it has lead to conducted in laboratories, thus it lead to the extra cost in oil palm production. The culling is performed to chop off the bad plantlet, also lead to the decline in yield the palm oil. Apart from that, tissue culture plays an important role in maintaining the same trait as its good parents. Plants as the explant are taken to conduct the technique thus lead the high yielding in palm oil. In contrast, the seed germination is caused the high-yielding oil kernel due to the crossing of DXP forming Tinera variety as it is the available for wet heat method during seed germination thus lead on the highest potential yielding of palm oil. But culling process lead to the deduction of the overall amount of the seedling during the seed germination stage will cause the decrease in yielding the oil extraction. As for the future development in oil palm propagation, the microspore culture is for the production of homogeneous lines without the need for add or more generation of recurring surfing as it is the case with traditional inbreed lines. Protoplast culture considered as the way to create hybrids between sexual incompatibility species as well as the potential source of genetic material. And last but not least is that the DNA transformation is where for the discovery and cloning of the desired gene, beside the discovery of promoter and regulator genes that regulate when where the gene is expressed and also the regeneration of complete plants. I think that's all from me. Thank you.